Hi everyone. Before we start with the topic of our talk, I want you to make a simple thought experiment. I want you to close one of your eyes and with your finger gently press on the eyelid of the opposite eye, like this. So you will see that the whole world in front of you shifts in the direction of your finger pressing. And just think about it. You know that the physical world that you see in front of you is not actually moving, but only your visual representation of the world. So what we actually see is not the world itself, but merely a representation of it through the interface of your vision, through communication of the information carried in the light that is reflected from the objects in front of you and reaches your retina and ultimately is interpreted by the visual systems of your brain to create this world that you see. So perhaps it is not that surprising that today you'll hear about the brain-computer interfaces and the brain and the human mind from a neuroscientist working in the multimedia systems department at the Faculty of Electronics, Telecommunication and Informatics of the Gdansk University of Technology. So I'll be talking to you, or should I say interacting with your senses of vision and hearing through this video, just teasing you a little bit, about the very thing that enables our communication with each other now and with the world around us, the human brain. Let's first introduce the object of this talk. The brain is by far the most complex object in the universe in terms of the number of possible connections between the billions of brain cells and trillions of connections between them. Mathematically speaking, the number of theoretically possible combinations of these trillion connections exceeds the number of atoms in the universe. So this theoretical number is not the actual number. The theoretical number of how many ways there are to connect those trillion connections is actually bigger than all the particles that make the universe. Yet these astronomical numbers are contained within approximately 1 kilogram of jelly that I can carry in the palms of my hand. But how can we interface with the brain? How can we see and hear its activity to understand what it is doing, to understand the complexity that is going on when the brain processes information? And can I communicate back with it to change or restore the activities that don't work in brain disorders? This is what I'll be talking to you about. So just like with our senses, the computers and special recording techniques that we use only see a small fragment of the spectrum of the brain's various activities. If you think about seeing again, your vision, we only see a tiny fragment of the spectrum of electromagnetic waves that are out there in the world, but we only see 1 or 2% of the visible light. So we don't see the infrared, for example, and the slower electromagnetic wavelengths. And likewise, we don't see the ultraviolet light and the faster electromagnetic waves, even though they are there. We just simply do not have ways or means to receive them. So here you can see a strip of polyimide electrodes, as well as some larger wide strip of round electrodes that record from small parts of the human cortex, just tiny parts, which are exposed surgically during an open brain surgery. Brain cells that are underneath those electrodes that record, they communicate information between each other through electrical energy. They speak the language of electricity. When the electromagnetic wave of light reaches the retina of your eye, or the mechanical energy of the air vibrations of sound reaches the cochlea of your ear, they are transformed into changes of electrical potential of the brain cells, also called neurons. And these electrical potentials are then sent and propagated as electrical impulses across networks of neurons in various parts of the brain. Then synchronized neuronal networks give rise to oscillations of the electrical field potential, which are commonly known as brainwaves. An example would be the famous alpha rhythm, which you might have heard about, which is present in the back of your brain with your eyes closed. And there are other slower oscillations, like the delta or theta rhythms, or the faster gamma rhythms. And we can record and study the language of these electrical activities by placing special electrodes on the cortical surface, just like you see here in the photograph, or implant them deep, penetrating into the brain. Of course, this is only possible in special cases of brain disorders that require implantation of electrodes through the skull to treat drug-resistant seizures or severe movement or mental disorders. Here in the upper corner, you can see an example of a grid of electrodes covering the side surface of the brain's cortex, as well as a straight array of electrodes penetrating deep into the sausage-like structures called the hippocampus. Below that, there are 3 traces of brainwave recordings, or you could say intracranial EEG, from selected contacts on these implanted electrodes. These brainwaves of particular frequency from the spectrum of those theta, alpha, gamma, and other bands can be detected just like the burst highlighted in red, or they can be displayed as the colorful spectrogram that you see in the background, with warmer colors indicating more activity at a given frequency in y-axis and time in x-axis as the patients are remembering stimuli presented on a computer screen. So this is one way in which we can visualize or translate what the brain is doing and what ultimately is created in our mind. In other words, we display with color to our eyes what happens in the head of the patient. You can think of it as a translated message of what happened in the mind, broadcasted to you by the media of a simple one-way brain-computer interface. But this is just a macro-scale picture of those brainwaves taken from far away and not seeing the details of what is going on underneath those waves. Another way is to look very carefully at individual impulses of specific neurons or at the local fast brainwaves generated by local assemblies of neurons that underlie emergence of a concept or idea in our mind. In this example, bringing a thought about a sleeping baby into mind is associated with specific neurons, here and there, lighting up with electrical energy which is captured as single impulses, known as the action potentials, by the nearby small golden microelectrode contacts, or as aggregated fast brainwaves, known as high-frequency oscillations, by the larger silver macroelectrodes. Notice that these local activities would be captured at different points along the length of this great depth electrode, going horizontally through the brain tissue, in this case. In a way, such electrodes can hear the microscale of very quiet whispers of the brain cells that take part in creating individual concepts or thoughts in our mind. They effectively provide an interface between the brain cells and the computer that reads their electrical activity. The same electrode contacts can also be used in the opposite direction, to deliver electrical current into the brain and stimulate the electrical activity of the brain cells. I'll tell you more about this a little later. So let's take a step back and realize what it really means. These unique recordings of electrical activity give us access to the cosmos of our mind. Our thoughts, memories, emotions can in principle be traced down to these electrical activities generated in distinct parts of the brain by specific brain cells. And the same activities, in turn, can be locally stimulated by the brain-computer interface through particular electrodes to improve abstract processes in our mind. Gosh, this is where the material meets the abstract, where the electrical impulses of the cell membrane meet abstract ideas in our mind. It is phenomenal. Just imagine how such brain-computer interfaces could be used to bring back people's memories, identity, or even consciousness that are impaired in many brain disorders. The key to accomplish this task, though, is to understand the language that the brain cells use to generate the contents of our mind, and to apply this knowledge to effectively communicate with them to restore or modulate their electrical activities. It sounds simple, but we still do not know how many cells support generation of a single thought, where exactly are they in the brain, and the electrical activities that are crucial for this process. You might have heard about the multi-billion projects for brain-mind interfaces, like the Neuralink from the inventor of Tesla cars, and other projects that are now, there's multiple of them around the world. They develop very impressive technology, but have little idea where to implant it. So where can we start? In the beginning was the Word, and the Word became flesh. This Bible verse from the Gospel according to John is very pertinent for this talk, because it links the most fundamental abstract concept, a word, with the material of the flesh, of the brain. The abstract, the word, and the brain, the flesh. Our brain and mind electrophysiology lab started with investigating how words are encoded and remembered in the brain. We knew that many different parts of the brain are electrically activated when words of specific meaning are displayed. For example, a word hammer would activate not only the visual parts located in the occipital cortex in the back of the brain, but also the hand movement areas in the motor cortex, which is located more in the front. Rose or sheep would likewise activate yet different areas, related more with the sense of smell, touch, emotion, or others. It becomes even more complicated with more abstract words like soul. So the problem is where to put the electrodes for the brain-computer interface to communicate with the contents of our mind. In this case, to read or stimulate memory for specific words in our mind, we cannot place the electrodes into all those various brain areas. But is there one area that controls remembering words in general? This is again this major obstacle for all the current brain technologies. And this is what we tried to address in an American project of a global neuroscience initiative that I was involved in before starting our laboratory at the Gdansk University of Technology. The goal of that project was to develop technology for a brain-computer interface to record and stimulate its activity for memory enhancement. In other words, we worked to develop a prototype of the world's first memory prosthesis. And you can see in the glass brain on the right of this photograph from an exhibition of the Museum of Science and Industry in Chicago. You cannot quite see it in this photo, but the prototype had a depth electrode implanted deep into the hippocampus, you remember the sausage-like structure, and the surrounding cortex inside the temporal lobe, so deep under the temples of your head, based on the idea that these brain areas control remembering words. Well, this exhibit has likely been changed because we found that using the brain-computer interface in these areas would actually impede memory and not make it better. A team of scientists that I was leading at the American Mayo Clinic as part of that project found another area called lateral temporal cortex, which is just beneath our temples, not deep down there. And that was the only stimulation target that improved remembering words. And this one discovery that we made with one Mayo Clinic patient, and then confirmed with 3 more patients, and then published in 2 scientific papers reporting this memory enhancement in overall almost 50 patients, completely changed the focus of the entire project and led to a startup company and establishment of the Brain and Mind Electrophysiology Lab now in Poland. So to help you realize the significance of this discovery, we will now make a quick experiment. In a moment, you'll see a video with this very patient as the discovery is being made. The patient is stimulating in this new target of the lateral temporal cortex for the first time. This is actually historical footage of this discovery being made. But you will do this task together with him. Your task is to remember as many of the 12 presented words as you can. The words will be presented one after another. The patient is also seeing them with you, but on his screen. The ones in yellow are indicated when electrical stimulation was applied. And unfortunately, I will not be able to stimulate your brain through this video now, so only he will be stimulated. After the last words, he will be doing some mathematical problems to distract him. Your job will be easier, to count how many words you can recall without any distraction. So once the last word is presented, you can start recalling and thinking, dog, woods, whatever, and count how many you remember. And then you will see how many did the patient remember and compare with your score. Are you ready? Okay, let's go.
Skunk, suit, snail, seed, cloud, juice, moth, plain, thread, dark, wood, sky.
No kidding. All of them.
There's a good picture in my head. A skunk wearing a suit.
So probably you're shocked because you remembered all 12. He remembered all 12 and you did less than 9, I guess, probably around 7. We were also shocked on that day in this room at Mayo Clinic because he had many more very good trials like this. This was not the only case. And it is astounding that he did not physically feel the effect of stimulation, but said that he could picture it well in his head. So just think for a moment about what happened there. The brain-computer interface tapped into his mental process of picturing the words. People can typically remember 5 or 6 items in this task in their working memory, and there is a limit for that you cannot remember more than 7 plus minus 2 items, whatever we try to remember, words or digits. On average, we have increased his abstract mental capacity to remember approximately 20% more than what his brain was physically capable of without the stimulation. We overcame the abstract limit of how many words he could remember by simply passing electrical current through a distinct part of the brain. What is even more exciting is that our latest findings suggest that there is another area in the very front of the brain that could have better effects still. So we know the areas that we can use for brain-computer interface. What we're trying to figure out now is what happens inside these areas with specific brain cells and how we can communicate with them. So could this allow us to read specific contents of the mind, i.e. specific words like dog or grandmother, and enhance remembering those that were likely to be forgotten? This brings me to the last component of the brain-computer interfaces that we are working on, tracking of eye movements. In contrast to the electrodes that we have to invasively implant into the brain, the eye tracking technologies are completely non-invasive and require only a small camera with infrared light and efficient computer algorithms to process the images recorded by the camera. What we found is that by tracking the size of pupils in your eyes, we can make a good prediction of whether a word is going to be recalled or forgotten. Why is it relevant for the brain-computer interfaces, for communicating with the mind? Well, we know that the pupil size reflects also the release of brain molecules called neurotransmitters that regulate many cognitive states, including attention. Knowing when the brain releases these neurotransmitters, like acetylcholine or noradrenaline, can inform the interface about one when it's not paying attention to remembering a word and is likely to forget it. An intervention like electrical stimulation could then rescue the memory for that word. This could in theory be entirely non-invasive, like many other brain-computer interfaces that I do not have time to cover in this talk. At least I will briefly mention to you an example of a toy using a simple non-invasive brain-computer interface. It measures brainwaves of particular frequency, those lower frequencies, from an electrode placed in a band over the forehead that you see in the photo. The power of these brainwaves are telemetrically sent to the device in front of the subject and changed into energy pumping airflow underneath a ball, so that it can be elevated. The higher the power of the brainwaves recorded, the greater the airflow elevating the ball higher. Now, what increases the power of the brainwaves over your forehead is the degree to which your mind can concentrate on this task. By concentrating your mind, you can synchronize large populations of brain cells so that the waves of electrical field potential are so powerful that they can be detected even non-invasively from the skin of your forehead. We don't have to go inside the brain to record them. We use this simple toy on most of our public events to show the principle of how all brain-computer interfaces work. This principle is applied to many clinical and industrial technologies, helping people to control things in the world remotely, using solely the power of the brain and mind. For example, a paralyzed patient can switch on light in a room or a television. Or patients with a robotic arm can control its movements by simply imagining these actions in their mind, and the robotic arm is changing them into mechanical movements of the robotic fingers. If you would like to try how it feels to control things in the world with your mind, visit one of our events by following our website or the Facebook page of the Brain and Mind Lab. So to summarize, we started with a thought experiment of moving your eye and the whole world together with it to show you the simplest way to interact with your mind. Then I told you how the contents of your mind can be recorded and translated from the language of electrical activities of the brain cells. I gave you some examples of some special electrodes that can provide an interface with specific brain cells that support processing of memories. I showed you our discovery made with a brain-computer interface that used electrical stimulation to successfully increase the ability to remember words in the mind. And you compared your performance without the interface with the patient's performance to convince you that these technologies can actually be very useful to restore brain functions or even enable computer-mediated control of the outside world. All these technologies harness the electrical activities associated with the processes of our mind. So can we go even farther to understand the origins of our mental activities? Let's think, where do our thoughts come from? Some are triggered externally by the things that we see in the outside world. You see a dog, you think of a dog. Whereas others are triggered internally from the deposits of your memory, of your past events, when, for example, you close your eyes. So is it possible to interface with these internal sources using the same brain-computer recording and stimulation technologies? If so, then maybe we can also study deeper sources of inspiration of our thoughts in the human soul or even outside of this world. There are multiple examples of mystics and visionaries across all ages who were receiving confirmed prophetic messages and revelations from the divine sources in their prayer. The most known examples include Saint Padre Pio, who regularly received information about people and events that he never encountered before, or the three uneducated children of Fatima, who during a series of Marian apparitions received information about the world events that actually came true years later. Even now, there are many astonishing examples of the so-called word of knowledge and prophetic images, when detailed knowledge about people is received without knowing them. So where do these words and images come from when they appear in the mind of a praying person? How do brain cells receive and generate the electrical activity that underlies these words compared to normally presented words, like for example in our memory task? And could this tell us something about our mind, soul, or even about God and their ultimate origin? So technically, these are not the questions that we are addressing in our brain and mind electrophysiology lab at the Gdansk University of Technology, at least not yet. But our research addresses the fundamental brain processes for these and other functions by using the brain-computer interfaces that use the electrical activities of neurons inside the human brain and the non-invasive signals recorded from our eyes. Our current goal is to use these to improve mental processes involved in memory and higher cognitive functions that are impaired in epilepsy, Parkinson's disease, or dementias like the Alzheimer's disease. Czadas Topciu and Dr. Michał Lech are the main researchers behind this project, which is funded primarily by the Foundation for Polish Science. They are both experts in applying computer technologies to solve clinical problems. Czadas worked previously with technologies applied for restoring functions in face and arm transplant patients, whereas Michał previously applied eye-tracking technologies to communicate with and assess cognitive states in patients suffering from disorders of consciousness. Both of them have also engaged in research at the American Mayo Clinic as part of our international network of the Brain and Mind Electrophysiology Lab. We currently have researchers from universities in 3 states in the USA and from 2 academic and clinical centers in the Czech Republic. Our group meets every Friday in different time zones to discuss the latest progress in the lab projects. Thanks to the recent government award for the Gdansk University of Technology for the best technical research university in Poland, we are now hoping to develop this international network of collaborations with the funding opportunities for the new BioTechMed Center at the university. These will prepare us to compete for the most prestigious European and American funding programs in the ongoing global neuroscience initiatives. By and large, this is a very favorable time to perform world-class neuroscience research in Poland, and specifically in Gdańsk. Last but not least, our research at the Brain and Mind Electrophysiology Lab would not be possible without the research, funding, and industrial partners listed here. We are very privileged to work in a team of the top clinicians, biomedical engineers, and computer scientists from the leading Polish, European, and American institutions. Special thanks to Professor Gregory Worrell at the Mayo Clinic, Professor Daniel Wójcik at the Nensky Institute of the Experimental Biology at Warsaw, Professor Paweł Tabakow at the Medical University of Wrocław, and our renowned Gdańsk researchers, Professor Andrzej Czyżewski and Professor Jarosław Sławek, without whom our laboratory would not have even started. We are very grateful for the visionary funding programs of the Foundation for Polish Science, which actually brought me back to Poland, and the National Agency for Academic Exchange, and the Polish Ministry for Science and Higher Education. And finally, we would not have the tools for our brain-computer interfaces if not for the companies that work with us to develop the next generation technologies to treat brain disorders. If you would like to stay tuned and know more about our latest discoveries or help promote our research, then visit our website or Facebook page. We regularly post news and videos from our various projects. You can also find us through the websites of our university, faculty, and the Multimedia Systems Department. Feel free to drop us an email, a question, or attend one of our outreach events. We are looking forward to hearing from you all, and may God bless you. Thank you for your attention. Goodbye.